Hello friends, I am Dr. Marathe from Department of Microbiology. Today I will explain another pathway and that is known as pentose phosphate pathway. Pentose phosphate pathway. As some pentoses are formed during this pathway, that's why this is also known as pentose phosphate pathway. No doubt, this is the oxidative pathway. This is the example of oxidative pathway like other pathways. But apart from the oxidation of glucose through this pathway, some important biomolecules are synthesized, compounds are synthesized. These compounds, they are like triodes, tetrodes, pentose, hexose and heptoses. All of you know that pentoses like ribose. So ribose, it is used for the synthesis of nucleotides like ATP, GTP, CTP, TTP, UTP, where the ribose sugar is required for their synthesis. This is the most important thing. And whatever the reducing power it is generated during this pathway, that reducing power it is used for biosynthetic reactions where the biosynthesis occurs. There, for reduction purpose, these reducing powers are used. This is the most important use. So that's why I am saying what? It is oxidative pathway, no doubt. But along with the oxidative pathway, this pathway, it has a great significance that it provides pentoses, reducing powers which are required for the biosynthetic reactions. So let us start this pathway. So, like other pathways, glucose, we will consider to understand how many carbon atoms are evolved, okay, in the form of carbon dioxide. So we must consider the 6 molecules of glucose. Okay, so 6 molecules of glucose. H, OH, H, OH, OH, H, H, OH. Here it is CH2OH, H. Now this is alpha B glucose. Now the reaction, it is converted into like a normal EMP pathway and other pathways. It is converted into glucose 6-phosphate. In this reaction, phosphorylation occurs and phosphate group is denoted by ATP. The name of the enzyme, all of you know that it is glucokinase, which requires magnesium ions for their activity. Okay, now it is converted into 6 molecules of glucose 6-phosphate, CH2OP. After phosphorylation, HOH, OH, OH, H, H, OH, H, OH. In the next reaction, what happens? There is a dehydrogenation, but this time the electron acceptor is NADP. So NADP plus, it gets reduced to NADPH plus H plus. So from where the 2 hydrogen atoms are removed. Okay, so I'll show this one and this one. These 2 hydrogen atoms are removed in the form of electrons. Okay, and the structure becomes like this. So here, due to loss of 2 hydrogen atoms, there is a formation of C double bond O. Okay, so H, OH, OH, H, H, OH, CH2OP and H. Okay, so due to the loss of 2 hydrogen atoms, this becomes a lactone ring. That's why the name of the compound, it is 6 molecules of 6-phospho gluconone delta lactone, 6-phosphoglucono delta lactone. Okay, so obviously it requires 6 NADP and we reduce to 6 NADP plus ester. Similarly here 6 ATP to 6 ADP. Okay, so every time you have to consider 6 molecules. Then is the further reaction by the enzyme lactonase, okay, which is accompanied by splitting of water molecule. It is accompanied by splitting of water molecule. Okay, so due to that, what happens? The ring gets opened. Ring gets opened from here. Ring gets opened. Okay, and whatever the H plus and OH minus ions are produced after the hydrolysis of water, so it forms CO and OH from the water. Then becomes COOH. Carbon number 1. From carbon number 2, HCOH. HCOH. Okay. Now carbon number 3, that is OH, CH. Now carbon number 4, that is HCOH. Okay. Then carbon number 5, HCO and 1 H plus from the water. Okay. So that becomes HCOH. And the carbon number 6, it is CH2OH. CH2OH. Now this is known as 6-phosphogluconate. 6 molecules of 6-phosphogluconate. This reaction is catalyzed by enzyme lactonase. Okay. Now in the next step, the further dehydrogenation reaction occurs. Okay, means oxidation occurs. And this is catalyzed by the enzyme 6-phosphogluconate dehydrogenase. Okay, so during the reaction again NADP acts as an electron acceptor. 6 molecules. They accept the electrons from the 6-phosphogluconate. Simultaneously, this is accompanied by loss of carbon dioxide. So, there is a loss of 6CO2. Okay, and the name of the enzyme is 6-phosphogluconate dehydrogenase. 6-phosphogluconate dehydrogenase. Okay, now how the CO2 it is lost, that we see. Okay, now this COO from the COH, it is liberated as a CO2. Okay, and 2 hydrogen atoms, they are removed from the carbon number 3. So after removal of 2 hydrogen atoms and carbon dioxide, what remains? Look at the structure. That is HCH. Means that is what? CH2OH. From this carbon, 2 hydrogen atoms are already removed. So only oxygen remains on this carbon. So that is what? C double bond O. Okay. Remaining structure as it is HCOH, HCOH and CS2OP. Okay. Now this structure is known as ribulose 5-phosphate. Ribulose 5-phosphate. 6 molecules. Now you can consider here there was a 6 carbon compound. 6-phosphogluconate. It is a 6 carbon containing compound. But after the reaction, what it is formed, that is a ribulose 5-phosphate, which is a pentose. Means 1 carbon it is reduced. Okay, 1 carbon is reduced. And how many molecules are there? 6. That means 6 molecules. Sorry, 6 carbon atoms are removed in the form of carbon dioxide. Okay, means there is a loss of 1 hexose. There is a loss of 1 hexose. Okay, so now this ribulose 5-phosphate, it's a pentose Now this can be used for another biostatic purpose. In the next reaction, it is catalyzed by two enzymes. One, it is known as ribulose 5-phosphate apimerase. What's its name? Ribulose 5-phosphate apimerase. So ribulose 5-phosphate undergoes the apimerization reaction, and its apimerase form, that apimerase is known as xylulose 5-phosphate. Okay, but out of 6 molecules of ribulose 5-phosphate, 4 are apimerized. Okay, so the structure becomes what? CS2OH, C double bond O. So apimer of this shows the change in the position of H and OH minus on the ribulose 5-phosphate. Okay, see, you can see the difference. That hydroxy group, it was there on the right side, now it is on the left side. This is known as xylulose 5-phosphate. Okay, so how many molecules? 2 plus 2 means 4 molecules of ribulose 5-phosphate. These are apimerized to 4 molecules of ribulose 5-phosphate. For our convenience, I have shown 2 plus 2. Later you will come to know why I have written like this. Okay, and remaining 2, these are isomerized. And which is the isomer of ribulose? That is ribose. Ribulose is the ketonic sugar, and ribose is ribulose is the ketonic sugar, and ribose is what? Aldehydic sugar. So remaining 2 molecules of ribulose 5-phosphate, they are isomerized to ribose 5-phosphate by the enzyme ribulose 5-phosphate ketoisomerase. Ribulose 5-phosphate ketoisomerase, and here it is ribulose 5-phosphate apimerase. Okay, now let us draw the structure of ribose. C double bond O H H C OH H C OH again H C OH, and last is what? CS2OP. Last is what? CS2OP. Now this is known as ribose 5-phosphate 2 molecules. Okay, so what happened during the reaction? Out of 6 molecules of ribulose 5-phosphate, 4 are apimerized to xylulose 5-phosphate, and remaining 2 are isomerized to ribose 5-phosphate. Okay, now up till this process, apimerization was there, isomerization was there, decarboxylation was there, dehydrogenation was there. Okay, so everything was there, means oxidation was there. Okay, but from this, the remaining pathway shows only interconversion of sugars. Okay, now how and which enzyme is involved in this process? Let us talk about that. Okay, now these 2 molecules undergoes the interconversion, but the enzyme is transketolase. Here it is called as a transketolase. I have shown in a short form TK, transketolase. So this transketolase transfers a 2 carbon fragment. I will show you 2 carbon fragment. Okay, so this 2 carbon fragment, it is shifted from xylulose 5-phosphate on ribose 5-phosphate. Okay, so ultimately it forms a 7 carbon sugar. That 7 carbon sugar is like this. Let us draw the structure of 7 carbon sugar. CS2H, C double bond O, here OH, C H, H, C OH, H, C OH, again H, C 1, 2, 3, 4, 5, 6. And last one is what? CS2O3. So this is nothing but what? 7 molecules of 6 molecules of pseudoheptulose. Pseudoheptulose 7-phosphate. This structure is known as pseudoheptulose 7-phosphate. This is predominantly found in one of the family of plants. Okay, this heptulose sugar that is predominantly found in some family of the plants. That's why the name is given pseudoheptulose. Okay, and remaining 1, 2, 3 means remaining structure remains. That is what? C, double one O, H, COH, and CS2OE. This is very familiar structure. Who has studied the glycolysis and other pathways? So that is what? Glycerol they have 3 phosphate, 2 molecules. So 2 molecules of pseudoheptulose 7-phosphate and 2 molecules of glyceraldehyde 3-phosphate. These are formed. You can simply understand nothing changed. Okay, only interconversion of sugar and carbon number remains same. Okay, so earlier you what? Xylulose, it is a pentose. How many molecules are there? 2. So 5 into 2, 10. Then ribose, it is also pentose. So how many molecules were there? 5 into 2, 10. 10 plus 10 carbon atoms were, means 20 carbon atoms were there. So let us check whether this number is conserved or not. So pseudoheptulose, it's a 7 carbon sugar. So 7 and 2 molecules are there. Okay, sorry, there should be 2 molecules of pseudoheptulose 7-phosphate. Means it's a 7 carbon sugar, so 7 into 2, 40. And glyceraldehyde 3-phosphate, which is a triose, and molecules are 2. So 3 into 2, 6. 6 plus 14, 20. So the carbon number is restored. Nothing changed. Okay, only a 2 carbon fragment, it is shifted from one structure to another structure. Next reaction, it is catalyzed by this time the name of the enzyme, it is trans-aldolase. I have written on the short form of this, that is trans-aldolase. Here it was trans-ketolase. Okay. Now what is the difference between trans-aldolase and trans-ketolase? Let us talk about that. So what happens? The trans-aldolase transfers the 3 carbon fragment. Okay. So it transfers this 3 carbon fragment from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate. And what is formed? Let us see. So the structure, it becomes remaining structure CC in 4 carbons. Okay, C double one O, H, COH, COH, and CS2OP. This is known as 2 molecules of erythrose 4-phosphate. Erythrose 4-phosphate. Okay, all of you know that erythrose, it's a 4 carbon containing sugar. So this is also known as tetrose. Okay, so it is an example of tetrose. And after shifting of these 3 carbon on this one, it becomes CH2OH, C double bond O. Okay, then OH, CH, then H, COH, then H, COH, and CS2OP. This is another familiar structure. If you have studied the EMP pathway, this is known as fructose 6-phosphate. 2 molecules are there. Fructose 6-phosphate. Okay, so 2 molecules of fructose 6-phosphate. Carbon number, it is reserved, is maintained. Okay, it is conserved. Okay, there is no change, only interconversion of sugar occurs. That is carried out by the enzyme trans-aldolase. So earlier there were 20 carbon atoms. Okay, now let us see whether these are conserved or not. So this is erythrose, 4 carbon, 4 into 2, 8, and 6 carbon, 6 into 2, 12, 12 plus 8, 20. Okay? Now in the next reaction, now let us talk about the next step. This next step is catalyzed by the same enzyme, that is transketolase. Transketolase, which is responsible for the removal of a 2 carbon fragment from xylulose 5-phosphate, and it is transferred on erythrose 4-phosphate. Okay, so we'll write here the structure. How many molecules of xylulose phosphate were there? There were 4 molecules of xylulose 5-phosphate. Okay, but out of these 4, 2 were already used. Now 2 are remaining. So these 2 xylulose 5-phosphate, xylulose 5-phosphate, that is CS2OH, C double bond O, OH, C, H, H, C, OH, and CS2OP. Okay, so these are the 2 molecules of xylulose 5-phosphate. During the reaction, the transketolase it acts or it catalyzes the reaction. So when it catalyzes the reaction, it transfers the 2 carbon fragment. So this 2 carbon fragment, it is transferred on this erythrose 4-phosphate. Okay, so the enzyme it is always transketolase. What is the enzyme? It is transketolase. So it forms another molecule of fructose 6-phosphate, CS2OH, C double bond O, OH, C, H, H, C, OH, and CS2OP. So 2 molecules of fructose 6-phosphate are formed. So how many carbon remains? Okay, 1, 2, 3. Okay, so this 3 carbon structure, it is known as C double bond O, H, HC, OH, CS2OH. So 2 molecules of G3P. Okay, now all of these are familiar structures of Embden-Mayer of Partners pathway. So G3P means glyceride 3-phosphate and fructose 6-phosphate. Okay, so these 2 fructose 2 molecules of G3 through the reverse of glycolysis, they are again converted into 1 molecule of fructose 6-phosphate. It is converted to 1 molecule of fructose 1,6-phosphate. So how many fructose 6-phosphate molecules are regenerated? So here 2 plus these 2, 4, and this one is 5. Okay, through the reversal of glycolysis, these fructose 6-phosphate molecules, 5 fructose 6-phosphate molecules, they are converted into 5 molecules of glucose 6-phosphate. So already during the initiation of pathway, during the starting of the pathway, we have taken 6 molecules of glucose 6-phosphate, but after this pathway, 5 are regenerated. Okay, that means 1 molecule it is removed in the form of carbon dioxide here during this process. So 6 carbon atom containing glucose 6-phosphate, it is removed, it is evolved, it is released in the form of carbon dioxide. Okay, so this is what the pentose phosphate pathway and its significance. I hope you must have understood this. Thank you very much.